Evolutions and behavior. Emergency of evolutionary thought. Origin of life. Abiogenesis. Abiogenesis is the generation of life from non-living matter. Abiogenesis is now more precisely known as a spontaneous generation. This theory states that a complex living organism are generated from decaying organic substances. Examples: organisms like mice spontaneously appear in stored grains, or maggots spontaneously appears in meat. Francisco Redi, an Italian physician who was the first to disprove the theory of spontaneous generation by performing a controlled experiment. In 1668, Redi performed an experiment to check whether the maggots really come from decaying meat. He did this by placing a meat in a few jars and covered half of them with a fine gauze while leaving the others uncovered. The maggots developed only on the meat in the uncovered jars. From this, they already concluded that the maggots did not come from the meat, but from the tiny eggs that flies had laid on the meat. Since the flies could not land on the meat in the covered jar, they could not lay eggs on that meat, and no maggots formed. Therefore, decaying meat could not produce maggots. Later, Lazzaro Spallanzani, an Italian naturalist, performed a similar experiment with broth. He put broth into 2 glass flasks and sterilized them by boiling the flask containing broth. One of the flasks was left open to the air. The other flask was sealed up to keep out any organisms that might be floated in the air. The organisms developed only in the uncovered flask. From this, the Spallanzani concluded that the microorganisms did not come from the broth but were or in air that enters the flask. Whereas in the sealed flask, the microbes do not develop. Unfortunately, many scientists were not convinced by this experiment. The Louis Pasteur, a French chemist, finally disproved the theory of spontaneous generation in the middle 1800. He performed the same type of experiment as Spallanzani. Louis Pasteur, however, allowed the air to enter the flask of sterile broth. He performed experiments with 2 flasks, one with a straight neck and other with the S-shaped neck. The flask with the straight neck allows both air and microorganisms to enter, whereas the other flask with the S-shaped neck allows only the air to enter but not microorganisms. The broth in the second neck flask becomes contaminated with microorganisms, but the broth in the flask with an S-shaped neck did not become contaminated. Therefore, Louis Pasteur shows that even though the air could get in the flask, the broth did not produce the microorganisms. The scientists finally were conceived that the living things, no matter how small, do not come from non-living things. The present theory of where living things come from is called biogenesis. This theory says that the living things comes only from other living things. For example, the mice comes only from mice. The microorganisms such as bacteria can only come from the other bacteria. Since spontaneous generation was now proved incorrect, many scientists began to wonder how life started on the earth. The Oparin and Halden attempted to answer this question. They proposed that life has arisen from simpler molecules of the lifeless earth under much different atmospheric conditions than exist today. However, instead of life arising suddenly as previously spontaneous generation theory proposed, the Oparin and Halden believed that it occurs only a very long period. Chemical evolution. Our current understanding of condition on prebiotic earth and the idea of a gradual chemical evolution towards the life were first proposed independently by A.I. Oparin and J.B.S. Haldane. The Oparin-Haldane model suggests that under the strong reducing condition theorized to have been present in the atmosphere of the early Earth between 4 to 3.5 billion years ago, the inorganic molecules would spontaneously form organic molecules, the simple sugars and amino acids. The Oparin argued that a primeval soup or primordial soup of the organic molecules could be created in an oxygen-less atmosphere through the action of sunlight. These would combine in every more complex ways until they formed coacervate droplets. These droplets would grow by fusion with the other droplets and reproduce through the fission into daughter droplets. Around the same time, in the year 1929, J.B.S. Haldane published a paper in which he proposed that the Earth's prebiotic oceans have been formed a hot dilute soup in which the organic compounds could have formed. In 1953, Stanley Miller, along with his graduate advisor Harold Urey, tested his hypothesis by constructing an apparatus that stimulated the Oparin Haldane early Earth. Miller-Urey experiment. The Miller-Urey experiment, a classical experiment, fundamentally establishes that the Earth's primitive atmosphere could produce the building block of life from inorganic materials. It establishes that the conditions that exist in the Earth's primitive atmosphere were enough to produce organic molecules like amino acids. They set up an experimental investigation into the molecular origin of life. They innovate the experimental design consisted of the introduction of molecules through to exist in the early Earth's primitive atmosphere into a closed chamber. Methane, hydrogen, and ammonia gases were introduced into a moist environment above a water-containing flask to simulate a primitive lightning discharge. The Miller supplied the system with electrical current. After a few days, the Miller observed that the flask contains organic compounds and that some of these compounds were the amino acids that serves as essential building blocks of protein. Using the chromatological analysis, he continued his experimental observation and confirmed the formation of amino acids like glycine, alanine, aspartic acid, glutamic acid, beta-alanine. The hydroxy acid and other organic compounds like urea, lactic acid, glycolic acid, succinic acid, and propionic acid. Thus, the Miller-Urey experiment demonstrated how some biological molecules, such as simple amino acids, would have arisen abiotically. That is through non-biological process under the condition similar to those of early Earth. Complex biological molecules and protocells. Miller and Urey demonstrated the formation of simple biomolecules, but further chemical evolutions would depend on the polymerization or condensation of these monomers into polymers, that is, formation of macromolecules like polypeptides, polysaccharides. For example, the nucleic acid bases can be synthesized under supposed prebiotic conditions. The adenine is formed by the condensation of hydrogen cyanide, a plentiful compounds of prebiotic atmosphere in reactions catalyzed by ammonia. The other bases have been synthesized by similar reactions involved HCN and hydrogen cyanide and water. The sugar have catalyzed by ammonia, NH3. The other bases have been synthesized by similar reaction involving hydrogen cyanide and water. The sugar have been synthesized by the polymerization of formaldehyde CH2O in reaction catalyzed by divalent cations, alumina or clays. It is probably no accident that these compounds are the basic components of biological molecules. They were apparently the most common organic substances in the prebiotic times. In 1950s, the Sidney Fox and co-worker University of Miami produces a synthetic peptide-like product of molecular mass between 4,000 and 10,000 daltons by heating a dry mixture of amino acids at 150 degrees centigrade to 180 degrees centigrade, which he called proteinoids, thermal proteins. Unlike the linear proteins produced by the cells, the proteinoids are branched polypeptide chains. The proteinoids exhibit several properties similar to biological proteins, including the catalytic activity and susceptibility to digestion by protease enzymes such as pepsin and trypsin. Origin of organized structure. If a complex molecules were produced from simpler ones, the macromolecules aggregates into membrane enclosed droplets, Koya servates and microspheres as a result of intermolecular forces, which exhibits some features of living systems such as organization, selective permeability, and energy use. Coacervate, meaning to assemble or cluster, is an aggregation of colloidal particles in a liquid phase that persist in the form of tiny droplets. The coacervates show a few interesting properties. One, able to exchange substances with the environment. Two, can increase in size. Three, selectively concentrate the compounds within them. Oparin has proposed that the coacervate may have been the intermediate between the loose molecules and the living systems. He has found that the droplets survive longer if they can carry out polymerization reactions. The microscopic membrane-bound sphere, termed microspheres, are formed when proteinoids are boiled in water and allowed to cool. The microsphere molecules aggregates of proteinoids are uniform size, stable, and double membrane bound that appears somewhat cell-like and can undergo fission and budding. Some cell-like properties such as osmosis, growth in size, selective absorption of chemicals have been ascribed to them. The spontaneous self-assemble of macromolecules into coacervates and microspheres indicates that the occurrence of similar structures under primitive conditions would probably give rise to more organized membrane-bound structure containing the molecules protocells. Later, the protocells that could reproduce would be acted on by natural selections, which would act as a creative force, successively enhancing the probability of advantageous molecules, reactions, and structure. Membrane defined the first cell. One of the crucial events leading to the formation of a first cell must have been the development of an outer membrane. The need of contaminant is easily fulfilled by a class of molecules that has the simple physiochemical properties of being amphipathic, that is, consisting of one part that is hydrophobic, water insoluble, and another part that is hydrophilic, water soluble. When such molecules are placed in water, they aggregate, arrange their hydrophobic portions as much in contact with one another as possible, and their hydrophilic portion in contact with the water. Amphipathic molecules of appropriate shape spontaneously aggregate to form bilayers, creating a small closed vesicles whose aqueous content are isolated from the external medium. All present-day cells are surrounded by a plasma membrane consisting of amphipathic molecules, mainly phospholipids. Presumably, the first membrane-bound cells were formed by spontaneous assembly of phospholipid molecules from a prebiotic soup, enclosing a self-replicating mixture of RNA and other molecules. It is not clear at what time in the evolution of biological catalysis and the protein synthesis this first occurred. In any cases, once RNA molecules were sealed within a closed membrane, they could begin to evolve in earnest as carry of genetic instructions. They would be selected not merely on the basis of own structure, but also according to their effect on other molecules in the same compartment. The outline of major steps postulated for the origin of modern cells. Gases of primitive earth gases, atmosphere, carbon monoxide, CO, ammonia, methane, hydrogen cyanide, and water. Heat or other forms of energy plus water to form amino acids. Then on heat, anhydrous condition to form proteinoids. Then on mild heat, it forms protocell. Then presence of water, replication of protocells. Then it forms energy storage, retrieval mechanism of ATP. And then it forms two types, the photosynthetic mechanism and internal protein synthesis. Then it forms then internal protein synthesis, it forms nucleic acid synthesis. The nucleic acid synthesis, the internal protein synthesis, again it forms energy storage in interval mechanism. The nucleic acid synthesis is again converted to gases of primitive earth atmosphere, like carbon monoxide, ammonia, methane, hydrogen hydrogen cyanide and water. Then the nucleic acid synthesis, it forms proto-ribosomes. The proto-ribosomes, it forms the genetic coding mechanisms. Then it forms contemporary cells. The photosynthetic mechanism, then it forms the proto-ribosomes, and the photosynthetic mechanism, it forms the genetic coding mechanisms. Current models. There is no true standard models of origin of life, but more currently accepted model built in one any way or another, depending on a few discovery about the origin of molecules and cellular components for life, which are listed in a rough order of postulated emergency. 1. The probability condition results in the creations of certain basic small molecules, monomers of life, such as amino acids, demonstrated in the Miller-Urey experiment by Sidney L. Miller and Harold C. Urey. 2. The phospholipid can spontaneously form a lipid bilayer, a basic component of cell membrane. 3. The polymerization of nucleotides into a random RNA molecules might have resulted in self-replicating ribosomes, the RNA world hypothesis. 4. The selection pressure for a catalytic efficiency and the diversity results in the ribosomes which catalyze the peptidal transfer. Hence formation of small protein, since oligopeptide complex with the RNA to form a better catalyst. Thus, the first ribosome is born and protein synthesis becomes more prevalent. 5. The protein outcompete ribosomes in catalyte ability and therefore becomes the dominant biopolymer. The nucleic acid are restricted to predominantly genomics. Theory of evolution. Biological evolution can be simply defined as a descent with a modification, which encompasses evolutionary changes at a genetic or organismal or population level. It is technically defined as a genetic changes in population of organisms that through time that leads to difference among them. The evolution helps us to understand the history of life. We can view evolution in two different ways, as a pattern of evolutionary change and as a process of evolution. The evolutionary theory deals mainly the how life changes after its origin. It is not a theory about the origin of life. The science does try to investigate how life started. Example, whether it happens near a deep sea vent, which organic molecules came first, etc. But these considerations are not the central focus of evolutionary theory. Regardless of how life started, afterwards it branched and diversified, and most studies of evolution are focused on those processes. Lamarckism. The Jean Baptiste de Lamarck, a French biologist, publishes a first truly mechanistic theory of evolution in his book Philosophie Zoologique, called the theory of use and disuse, or the inheritance of acquired characters. According to this theory, 1. The ecological condition in which an organism lives causes it to have certain needs. 2. These needs are met by modification of old organs or by production of new rudimentary organs. 3. By continual use of these rudimentary structures, they increases in size and functional capacity, and by disuse they may be degenerated and become lost. 4. The character changes brought out by the environment during the life of an individual becomes hereditary and thus can be transmitted to the next generation. Example, the Lamarck would say that the cavy fish are often blind because their ancestors have lived for several generations in dark conditions, and in each generation the eyes has progressively degenerated through lack of use. The long neck of modern giraffe could also be explained by Lamarckian theory. During time when food was scarce, the short-necked ancestors need to stretch their neck to get the leaves higher up in the trees. In this way they stretched their neck. In each generation, the exercising of neck in this fashion causes the increment in the length of the neck, which becomes genetically fixed. The Lamarckian theory may sound plausible, but it is unsupported by any unequivocal scientific evidence. Darwinism. The Charles Robert Darwin, 12th February 1809 to 1819, April 1882, was the English naturalist who proposed and provided the evidence for the scientific theory that all the species have evolved over times from one or a few common ancestors through the process of natural selection. This theory becomes widely accepted by the scientific community in the 1930s and now forms the basis of modern evolutionary theory. The Darwin compiled evidence for evolution by the natural selections for about 20 years. Between 1842 and 1844, he wrote a 230 pages essay summarizing his theory and the evidence for it. In the 1850, he began working on a detailed, the multi-volume book to present his theory to the scientific community. The Darwin might never have completed the book if another British scientist, Alfred Russel Wallace, 1823 to 1913, had not come up with the same idea in 1858. The Wallace formulated his theory and wrote it in an essay, which he sent a Darwin. The Darwin's fellow scientists persuaded him to let them present his theory and Wallace's essay jointly at a scientific meeting. The presentation excited very little attention according to the modest Darwin. However, the publication of Darwin's book of Origin of Species in 1859 changed biology forever. The Darwin clearly and logically presented the idea that natural selection is the mechanism of evolution. In Darwin's own lifetime, many scientists became convinced that evolution occurs by means of natural selection. Today, this theory is the unifying one for all biology. Darwin's theory of evolution by natural selection. The first, Darwin states that the variation exists among the individual of species. The second, he stated that the scarcity of resonance in a burgeoning population would lead to competition between individuals of the same species because all use the same limitation resource struggle for existence. Such competition would lead to the death of some individuals while others would survive. From this reasoning, Darwin concluded that the individual having advantageous variations are more likely to survive and reproduce than those without the advantageous variations survive to be the fittest. Analogical argument for natural selection. The fact one, all the species have such great potential fertility that their population size would increase exponentially if all the individuals born would again reproduce successful. Fact 2. Except for a minor annual fluctuation and occasional major fluctuations, the population normally displays the stability. Fact 3. The natural resources are limited. In a stable environment, they remain relatively constant. Fact 4. No two individuals are exactly the same. Rather, every population displays enormous variations. Fact 5. Much of this variation is heritable. Inference. Since more individuals are produced, then they can be supported by the available resource, but the population size remains stable. It means that there must be a fierce struggle for existence among the individuals of a population, resulting in the survival of often a very small part of the progeny of each generation. Interference 2. The survival in the struggle for existence is not random, but depends in the part on her hereditary constitutions of the surviving individuals. This unequal survival constitutes the process of natural selection. Inference 3. Over the generation, this process of natural selection will lead to continuing gradual changes of populations, that is the evolution, and to the production of new species. The Darwin coined the term natural selection to describe the process by which the organisms with favorable variations survive and reproduces at a higher rate, and inheritable variations that increases an organism's chance of survival and environment is called an adaptations over many generations, and adaptation could spread throughout the entire species. In this way, according to the Darwin, the evolution by natural selection would occur. Summary: the Darwin's theory invariability have a combination of some of the following 5 theory in mind. 1. Evolution as such. This theory states that the world is not constant or recently creates nor perpetually cycling, but rather is steadily changing, and that organisms are transferred in time. 2. Common descent. This theory considers that every group of organisms descended from a common ancestor, and that all group of organisms, including animals, plants, microorganisms, ultimately go back to a single origin of life on Earth. 3. Multiplication of species. This theory explains the origin of enormous organic diversity. It postulates that the species multiply either by splitting into daughter species or by budding, that is, by the establishment of a geographical isolated founder population that evolves into new species. 4. Gradualism. According to this theory, the evolutionary change takes place through the gradual change of populations and not by the sudden productions of a new individual that represents a new species. 5. Natural solutions. According to this theory, the evolutionary changes come about through the abundant productions of genetic variations in every generations. The relatively few individuals who survive owing to a particularly well adapted combinations of inheritable characters give rise to the next generation. Darwinism. The Darwin proposed his theory of evolution by natural selection without the knowledge of the source of variations in the populations. After Mendel's law were rediscovered and the genetic variations was shown to be generated by the mutation, the Darwinism and Mendelism were used as a framework of what came to be called the synthetic theory of evolution or Neo-Darwinism. According to this theory, although the mutation is recognized as the ultimate source of genetic variations, natural, that is positive selection, is given the dominant role in shaping the genetic makeup of population and in the process of gene substitution. Galapagos Island and Darwin's finches. The Galapagos Island, named after the large tortoises that inhabits them, or an archipelago of volcanic island distributed around the equator in the Pacific Ocean west of continental Ecuador. An archipelago, sometimes called an island group, is a chain or cluster of islands. The Galapagos archipelago is a cluster of 29 islands and islands of different sizes lying on the equator. The islands are all relatively young in geological terms, and they have never related to the adjustment maintenance of South Africa or with any other source area. All the organisms that occurs on these islands have reached there by crossing the sea as a result of chance dispersal in water, by wind, or by transport via another organisms. The Darwin's finches are a group of 14 species of small bird, 13 of which occurs on the Galapagos Island. This group of birds provided one of the most striking and best studied examples of evolution on the island. The scientists think that the Galapagos finches descended from an ancestral species that originally inhabited the mainland of South America. Since the Galapagos is a group of islands, the small flocks of bird could occasionally reach a new island where no other land birds were present. On these new islands, the immigration would become adjusted to the local conditions through the process of natural selections. The descendants of original finches that reaches the Galapagos Island now occupy many kinds of inhabitants on the island. In doing so, they would have become progressively more different from the original populations. The most obvious difference between these finches relates to the dietary adaptation and is reflected in the size and shape of their beaks. The finches of Galapagos Island provides an examples of adaptive radiations, the formation of new forms from an ancestral species, usually in response to the opening of new habitats. Among the 13 species of Darwinism finches that inhabits the Galapagos, there are three main groups: ground finches. These are six species of ground finches. Most of the ground finches feeds on seeds of different sizes. The size of their beaks is related to the size of the seeds on which the birds feed. Tree finches. These are six species of tree finches belonging to several diverse groups. These species also differs from one another primarily in beak size and shape, which reflects the adaptation to their food. The four species have beaks that are suitable for feeding on insects. The one of the largest beak feed on larger insects, and those three with smaller beaks feeds on a smaller insects. One of the three finches has a parrot-like beaks. It feeds on buds and fruits in the trees. The sixth species of the tree finch, the woodpecker finch, has a chisel-like beak. This unusual bird carries around a twig or a cactus spine, which it uses to probe for insect in deep service. It is an extraordinary example of bird that uses a tool. Warbler finches. The unusual bird differs from the ground finches and the tree finches in its habit and morphological adaptation. The warbler finch plays the same ecological role in the Galapagos wood that warblers play on the mainland, searching continually over the leaves and branches for insect. It has a slender warbler-like beak. Evidence of evolution. The evidence supporting the evolution model came from multiple distinct areas of biology and geology. These pieces of evidence can be grouped into four categories. 1. Direct observation of evolutionary change. 2. Homology and development. 3. Vestigial traits. 4. Fossil records and geography. Direct observation of evolutionary change. The small scale evolution can be observed in nature or generated experimentally in the laboratory. The classical story of the peppered moth in the Britain during the Industrial Revolution is an example of microevolution. The Grant and Grant have recorded evolutionary change in beak shape within and among the population of Darwin's finches over a period as short as 2 years. We can also drive genetic changes in laboratory population of Drosophila and other model organisms in the laboratory. By selecting certain phenotypic traits, we can generate the directional changes in the characteristics such as abdominal bristle number, lifespan, and avoidance to certain chemicals. The artificial selection experiments are also the foundations of agricultural improvement over the past 10,000 years. Homology and development. One of the most compelling lines of evidence supporting the common ancestry of species is that the similar structural element across the functionally diverse form. It becomes evidence from the comparative anatomy. We find the same bones in many different types of animals, but these bones are often modified to do different things. The hopping legs of the frog contains same bones as our own leg, but the frog's leg are highly modified to fulfill a different functions. Hopping. The wings of the birds and forelimbs of a bat contains the same bone as the arms of the human, but the size, shape, and even internal structure of these bones are all adapted to play different roles in each animal. We call structures like the wings of the bird and forelimbs of the bat homologous structure. The homologous structure or the structure that are derived from from a common ancestor. Even if they are superficially different, they are developmentally related. The homology does not mean that these structures must share the same function. The characters shared between the two species but not present in their common ancestor is called homoplasy. But very often in nature we find structures that are superficially similar, even though the organisms are completely unrelated to one another. These structures may even serve the same functions, like flying, and described as a analogous structure. The wing of the birds and the wing of the insect are good examples of analogous structure. In every physical and biological way, these wings are radically different from one another. One is a flat plane of exoskeletal material, and the other is the chordate forelimb shaped into an airfoil with hollow internal bones and an outer covering of the feathers. But they are both can be used to fly. The observations of structural homology supports the casual hypothesis of evolutionary homology that the common structural organization results from the common ancestors. The evidence of analogous structure further supports the evolutionary homology concept, as they indicates that the design is not necessarily constrained. That is, there is more than one way to build a functional part. In many cases, during embryonic development of organisms, the embryo exhibits the characteristics of embryos of its ancestors. For example, early in the development, the human embryo possesses gill slits like fish, and later exists a tail, the vestige of which present in adulthood as a coxswain at the end of the spine. These relict development forms suggest strongly that our development has evolved. Vestigial structure. Many species shows structural or genetic features that serves no apparent functions. The obvious examples of these individual: the presence of pelvic bones in snakes and whales, rudimentary eye in some cave fish species, presence of the nictitating membrane in humans, and the presence of pseudogenes throughout the genome of eukaryotes. These structures suggest strongly that the species has changed over time and descent from an ancestor with different ecological or genetic characteristics. A relevant phenomena suggested descent with modification is evidence of poor engineering or inefficient design is some characteristics of organisms. For example, the gene regulation is often a very inefficient process with multiple, often conflicting stages. These observations suggest that the existing structure are often co-opted during the evolutionary change to serve new functions. That is, there is descent with a modification as opposed to independent design of each species. Fossil record and biography. The fossils or the preserved remains or traces of organisms from the distant past. The fossil records remains first and foremost among the databases that documents changes in the past life of the earth. The sedimentary rocks are the richest source of fossils. As a result, the fossil record is based primarily on the sequence in which the fossils has accumulated in the sedimentary rock layers called strata. The fossil record shows that there has been a great changes in the kinds of organisms that dominate life on the earth at different points in time. The fossil record of most major animals group begins in the Cambrian. Biogeography is the science that explains the distribution of species and higher taxa on the surface of the earth. The geographic distribution of species may be endemic. The species that are limited to a particular area, cosmopolitan species that are found on all contained of globe, or disjunct species that are not confined to a single area but are distributed in more than one region with a gap between them. The biogeographic distribution of species supports evolution. For example, approximately 2000 species of the flies belonging to the genus Drosophila are now found throughout the world. About one quarter of them lives only in Hawaii. Similarly, more than 1,000 species of snails and other land mollusks are found only in Hawaii. The biological explanation for the multiplicity of related species in remote localities is that such great diversity is a consequence of their evolutions from a few common ancestors that colonized an isolated environment. The Hawaiian island are far from any mainland or other island, and based on geological evidence they never have been attached to other lands. Thus the few colonizers that reached the Hawaiian island found many available ecological niches where they could over numerous generations undergoes the evolutionary changes and the diversifications. Natural selection. The selection is a composite of all forces that causes a differential survival and differential reproduction among the genetic variants. When the selective agencies are primarily those of human choices, the process is called artificial selection. When the selective agents are not those of human choice, it is the natural selection. The natural selection is the non-random process by which the biological trait becomes common in a population as a function of the differential reproduction of their barrier or differences in the rate of survival. The natural selection can act on any heritable phenotypic trait and operate among any entities that reproduces, shows inheritance of their characteristics from one generation to the next, and vary in the fitness. The natural selection is the mechanism that drives evolution. Some evidence of natural selection has been seen in nature, but not to an extent that would change a species in any meaningful way. The evidence of natural selection, industrial melanism. The industrial melanism is a phenomenon used to describe the evolutionary process in which the initially high-colored organisms population become dark as a result of natural selection. The industrial melanism affects over 70 species of moths in England. It has been best studied in the peppered moth, Biston betularia. Prior to 1800, the typical moth species had a light colored pattern. The dark colored or melanic moth were rare. During the Industrial Revolution, soot and other industrial waste darkens the tree trunk and killed off lichen. Because light colored peppered moth relies on camouflage to avoid predation. These sudden changes in the environment made them highly vulnerable to predators. In course of time, the light-colored moth becomes rare and the dark-colored moth becomes abundant. The cause of this change was though to be selective predation by birds with favored camouflage coloration in the moth. By 1886, the dark-colored melanic were far more common, illustrating the rapid evolutionary change. Evolution of drug-resistant HIV. The evolution of drug resistance in HIV also provides an example of natural selection. The researchers has developed numerous drugs to combat this pathogen. The drug 3TC, lamivudine, is one of them. It is a nucleoside inhibitor and acts as a molecular analog of cysteine. When reverse transcriptase places a 3TC molecules instead of C, it is a replicating DNA chain. Chain elongation is stopped and the replication of HIV is stopped. The 3TC lamivudine, a resistant variant of HIV carrier, slightly different version of reverse transcriptase that are discriminate between the drug and the normal C nucleotide. The virus that carries these genes have no advantages in the absence of 3TC lamivudine. In fact, they replicate slowly than those they carry the most common forms. But once 3TC is added to their environment, it becomes a powerful selecting force favoring reproduction of resistant individuals. Here, the drug does not create resistant HIV. It selected resistant HIV that was already present in the population. The increase in the frequency of drug resistant HIV is almost certainly driven by natural selection. Modes of natural selection. If an individual has some form of trait that gives him a better chance of survival in each environment, evidently his offspring inherit the trait. This is known as a differential survival. The differential survival would refers to the survival of individual in an environment to which they are well adapted. The adaptation are genetically controlled by heritable. The differential survival leads to differential reproduction of allele responsible for the survival of next generation. The natural selection means differential reproduction among the members of the same gene pool. The natural selection can alter the frequency of heritable trait in three ways. These three modes of natural selection are: 1. Directional selection, 2. Stabilizing selection, 3. Disrupting selection. Under directional selection, the individual at one end of the frequency distribution do especially well, and so the frequency distribution of the trait in the subsequent generation is shifted from where it was in the parental generation. In nature, the directional selection can occur when one of the phenotypic extremes becomes selected for or against, usually as a result of changes in the environment. The industrial melanism provides an examples of directional selections. Natural selection. The selection is a composite of all the forces that causes differential survival and differential reproduction among the genetic variants. When the selective agencies are primarily those of human choice, the process is called artificial selection. When the selective agents are not those of human choice, it is called the natural selection. The features of directional selection. 1. Occurrence due to the change in environment in a direction. 2. Favors the phenomena which is not non-average or extreme. 3. Alters the mean value of the trait in the population in one direction. 4. Favors accumulation of those mutations that increases fitness in the changing environment. And 5. Elimination of normal or average individuals. Under stabilizing selection, the extreme variations from both ends of frequency distributions are eliminated. This type of selection tends to favor intermediate types. At the genetic level, the stabilizing selection acts to keep a population well adapted to its environment. In this situation, the individual closer to the average for a given trait will have higher fitness. A real life example is that of birth weight of human babies. The features of stabilizing selections. 1. Operates in the constant or unchanged environment. 2. It keeps a population genetically constant. 3. Favors the average or normal phenotype. 4. Introduces homozygosity in population. And 5. Checks accumulation of mutation. Under disruptive selection, both extremes are favored at the expense of intermediate varieties. It can be viewed as its opposite of stabilizing selection because the intermediate types are selected against. A clear example is provided by the different color patterns of the butterfly Papilio-Dardanus. The features of disruptive selections. 1. Previously homologous population breaks up into several different adaptive forms. 2. Extreme values have highest fitness and intermediate or mean value or relatively disadvantageous. 3. Occurs when a population previously adapted to a non-homologous environment is subjected to divergent selection pressure in different parts of its distributional area. Sexual selection. Adapting to the natural environment is only one way organisms can increase their ability to leave more offspring for future generation. Another method is to improve their ability to secure mates through the action of sexual selection, a form of natural selection. The sexual selection arises from difference in reproductive success caused by the competition over mates. Because variance in reproductive success can often be quite high, especially for males, the adaptation to the mating environment can sometimes be even more effective in promoting changes than adapting to the natural environment, and has resulted in the rapid divergence of triad resulted into reproduction. The sexual selection can result in sexual dimorphism, marked difference between the two sexes in secondary sexual characteristics which are not directly associated with the reproduction or survival. These distinctions include difference in size, color, ornamentations and behavior. For example, peacocks and peahens shows extreme sexual dimorphism. There is a sexual selection when the female chooses amongst the showiest males. The pressure for sexually reproducing organisms to have compatibility reproductive system ensures that male and female reproductive system will co-evolve. Pattern of evolution: convergent evolution and divergent evolution. We often find unrelated animals converging on the same form or structure because that form is very adaptive in their common environment. This special case of evolution is called convergent evolution. It describes the acquisition of the same biological trait in unrelated species. Examples of convergent evolution is the streamlined shape of sharks and dolphins. One is a fish, the other is a mammal, and they are related to one another in only the most distant sense. But if its life depends on swift movement through the water, then a streamlined shape is pretty much essential. Another example is wing in birds, bats, and flies. The convergent evolution produces analogous structure in contrast to homologous structure, which have a common origin. Similarly, among unrelated organisms, as result of convergent evolution is called homoplasy. The wings of birds, bats, and insects are homoplastic feature because the common ancestor of these three groups lacked wings. The spines, modified leaves, and thorns, modified stems are examples of homoplasty among plants. Spines and thorns look similar and both provide protection to plants, but the plants to which they belong are distinctly related. The homoplastic similarity can be anatomical, physiological, or behavioral. Divergent evolution. The divergent evolution is the process in which two or more related species becomes more and more dissimilar. In this case, the similarity is due to the common origin. The divergent evolution produces homologous structure. A good example of divergent evolution is Darwin's finches, which now have over 80 varieties which all diverged from one original species of finch. Another example of divergent evolution is the organisms have 5-digit pentadactyl limbs like the humans, bats, and whales. They all have evolved from a common ancestor. Coevolution. The term coevolution is used to describe cases where two or more species reciprocally affects each other's evolution. For example, an evolutionary change in the morphology of plants might affect the morphology of an herbivore that eats the plant, which in turn might affect the evolution of plant, which might affect the evolution of a herbivore, and so on. The co-evolution is likely to happen when different species have close ecological interactions with one another. These ecological relationship includes: 1. Predator, prey, and parasite or prey. 2. Competitive species. Third, mutualistic species. The plants and insects represent a classic case of co-evolution, one that is often but not always mutualistic. Many plants and their pollinators are so reliant on one another, and their relationships are so exclusive that biologists have good reasons to think that the match between the two is the result of a co-evolutionary process. But we can see exclusive matches between the plant and insect even when the pollination is not involved. Some General American Acacia species have hollow thorns and pores at the bases of their leaves that secretes nectar. These hollow thorns are the exclusive nest site of some species of ants that drinks the nectar. But the ants are not just taking advantages of the plant; they also defend acacia plants against herbivores. This system is probably the product of co-evolution. The plants would not have evolved hollow thorns or nectar pores unless their evolution had been affected by the ants, and the ants would not have evolved the herbivore defense behavior unless their evolution had been affected by plants. Parallel evolution. The parallel evolution is the development of a similarity in separated by the related evolutionary linkages through the operations of similar selective factors on both lines. The parallel evolution is similar to convergent evolution in that similar adaptation arises in different species. The main differences is that in parallel evolution the two species have a relatively recent common ancestor. Adaptive radiation. The natural selection can ultimately lead to the formation of new species. Sometimes many species evolved from a single ancestor species. The similarity in skeletal and muscular structure of a Hawaiian honey creeper led scientists to conclude that the 23 species of honey creepers had evolved from one ancestor species, such as evolutionary patterns in which many related species evolved from a single ancestor species, and it is called as adaptive radiations. The adaptive radiations most commonly occurs when a species of organisms successfully invade an isolated region where a few competition species exist. If new habits are available, the new species will evolve. Another interesting example in which one species give rise to multiple species that exploits different niches is the finches of Galapagos Island. There are 14 species of finches on the Galapagos Island, each filling a different niche on different island. All of them evolved from one ancestral species which colonized the island only a few million years ago. The ancestral finch was ground were dwelling seed-eating finch species and speciation. The concept of species. The species are the basic taxonomic unit of biological classification. This is a grouping of organisms of like kind into discrete and stable unit. There is a lack of conscious in how to define the term and what constitutes a species. Among several definitions of species, the most commonly used is the biological species concept. According to the biological species concept put forth by Mayer, the species are group of interbreeding natural population that are reproductively isolated from other such groups. The members of the species actually or potentially interbreed in nature, not according to similarities of appearance. Although the appearance is helpful in identifying species, it does not define species. When two organisms breed within a species, their genes passes into their combined offspring. As this process is repeated, the genes of different organisms are constantly shuffled around the species gene pool. The shared gene pool gives the species its identity. The strength of the biological species concept is that it gives emphasis on reproductive isolation. However, the number of species to which this concept can be usefully applied is limited. It does not apply to organisms that reproduce asexually all or most of the time, such as prokaryote. Furthermore, in biological species concept, the species are designated by the absence of gene flow. There are, however, many pairs of species that are morphologically and ecologically distant, and yet gene flow occurs between them. Further, natural selection can cause such species to remain distinct despite gene flow. Because of limitation of biological species concept, the alternative species concept are useful in certain situations. Phenetic species concept. According to the phenetic species concept, a species is a set of organisms that are phenetically similar and look different from other sets of organisms. According to this, phenetic similarity is all that matter is recognizing separate species. For example, if two frogs look the same even though they are prevented from mating with each other, they would be considered the same species according to the phonetic species concept. The classical version of the phonetic species concept is the typological species concept. The term morphological species concept is also used to refer the same concept. Phylogenetic species concept. The phylogenetic species concept essentially defines a species as a group of organisms whose members are descended from a common ancestor and who all possesses a combination of certain defining or derived trait. Hence, this concept defines a species as a group having a shared and unique evolutionary history. Evolutionary species concept. An evolutionary species is a group of organisms that shares an ancestor, a lineage that maintains its integrity with respect to other lineage through both time and space. At some point in the progress of such a group, the members may be diverge from one another. When such a the divergence between sufficiently clear, the two populations are regarded as a separate species. According to the Simpson, who proposed the evolutionary species concept, a single lineage of ancestor descendant population which maintains its identity from other such lineage and has its own evolutionary tendency and historical fit. Reproductive isolation. The reproductive isolation is an isolating mechanism that prevents gene exchange among the population. There are many barriers to reproduction that evolves during the course of speciation, which limits gene flow between the sister species. Each of the species may have its own countership display or breeding seasons, so that the members of the two species do not have the opportunity to interbreed, or the two species may be unable to interbreed successfully because of the failure of the eggs to become fertilized or to develop. This suggests a simple and a useful dichotomy between the pre-mating or pre-zygotic, that is pre-zygote formation reproductive isolation mechanism, and post-mating or post-zygotic isolation mechanism. Pre-zygotic isolating mechanism. The pre-zygotic isolating mechanism leads to reproductive isolation by preventing the formation of hybrid zygote. Ecological isolation. The species occupy different habits. Example, the two species of greatest shock in the genus Tamnophis occurs in the same geographic area, but one lives mainly in the water while other is primarily terrestrial. Temporal isolation. The species breed at different times. Example, in North America, the geographic range of the eastern spotted skunk, the Spilogale Putorius, and the western spotted skunk, Spirogale gracilis, overlap, but Spirogale Putorius mates in the late winter and Spirogale gracilis mates in the late summer. Behavioral isolation. The species engage in distinct courtship and mating rituals. Example, blue-footed boobies, inhabitants of Galapagos, mate only after the courtship display unique to their species. The part of the script called for the male to high step, a behavior that calls the female attention to his bright blow feet. Mechanical isolation. Interbreeding is prevented by structure or molecular blockage of the formation of zygote. The mechanism includes the inability of the sperm to bind to the egg in animals or female reproductive organs of a plant, preventing the wrong pollinator from landing. Example, even in closely related species of plants, the flower often have distinct appearance that attracts different pollinator. The two species of monkey flower, Mimulus, differ greatly in the shape and color of their blossoms. Thus, the cross-pollination between the plant does not occur. Post-zygotic isolating mechanism. The post-zygotic mechanism are those in which the hybrid zygote fails, developed abnormally, or cannot self-reproduce and establish viable population in nature. Hybrid inviability. Development of zygote proceeds normally, but hybrid does not survive. Example, some salamander subspecies of genus Ensatina lives in the same region and habitat where they may occasionally hybridize, but most of the hybrids do not complete development, and those that do not frails. Hybrid sterility. The hybrid is healthy but sterile. The mule, the hybrid offspring of a donkey and mare is sterile. It is unable to produce a viable offspring. The hybrid is healthy and fertile, but less fit, or infertility appears in the late generation, as witnessed in the laboratory cross of a fruit fly, where the offspring of the second generation hybrid are weak and usually cannot produce a viable offspring. Example, the hybrid offspring of donkey and horse and mule is robust but sterile. Speciation. The speciation is a lineage splitting evolutionary process that produces two or more separate species. It refers to the creation of new and distinct biological species by branching off from the ancestral population. It occurs when the gene flow is reduced sufficiently between sister population to allow each to become irrevocably committed to different evolutionary lineages. Reduced gene flow probably plays a critical role in speciation. The various modes had been presented whereby a single evolutionary lineage splits into two or more genetically independent lineages due to reduced gene flow. The mode of speciation are often classified as allopatric, parapatric, and sympatric speciation. Allopatric speciation. In 1963, the Ernest Mayr conducted that the speciation takes place through allopatry or geographic isolation. The allopatric speciation, allo means other, patrick means place, is the genetic divergence permitted by the geographic isolation. This process is dependent on random mutation, which accumulates steadily after a population had been subdivided into space. This subdivision can take place because of a geographic barrier such as a mountain range and water bodies. Because of two subpopulations are then reproductively isolated, each of the population accumulates in different mutations, and the two populations diverge. This divergence is the strongest where the population size of one or both subpopulation is small, because sampling error is high in the smaller population. These differences will eventually lead to reproductive incompatibilities that will be keeping the population distinct. One will know examples of allopatric speciation involve divergence of a Drosophila population in the Hawaiian archipelago. The reproductive isolation mechanism usually originate incidentally in speciation process, that is, they arise as incidentally during the process of evolution in isolated population rather than being selected for. When isolated population comes together again, the incomplete isolation mechanisms may allow hybrid to form. If the hybrid are normal, viable, and can freely interbreed with individuals of each parent population, then no speciation has been taken place. However, if the hybrid are at a disadvantage, the natural selection may favor stronger isolation mechanism. In this case, the organisms that mate with individuals from the other population leaves fewer offsprings. The result is a more effective barrier to hybridization. The region in which previously isolated population come into contact and produce hybrid are called hybrid zones. A simplified flow chart of allopatric speciation. 1. The single interbreeding population. There is 2. Geographic isolation between the population, and 3. Increased genetic divergence among geographic isolated population by means of genetic drift and selection, and 4. Selection for increased reproductive isolation, and for 5. Speciation completed. Sympatric speciation. The sympatric speciation, symp means same and patric means place. The sympatric speciation conversely does not require the geographic isolation. Instead, it relies on development of reproductive isolation mechanism to allow divergence of the two populations. This reproductive isolation can be results of a few factors, including competition for resource, disruptive selections, and sexual selections. In sympatric speciation, the selection acts against individuals of an intermediate types, either through decreased viability or decreased fecundity. This creates the evolution of mating preference and other mechanisms that results in decreased hybridization. In sympatric speciation, the selection enhances traits that promotes the divergence of the population. This is fundamentally different from allopatric speciation, where mutation passively leads to difference that allows divergence of population. Probably the best known examples of sympatric speciation is the divergence of Rhagoletis Pomonella, the maggot fly. This species has recently diverged into two subspecies due to the introduction of a apple tree in the northeastern United States, where hawthorn tree were native. Initially, the flies used the hawthorn fruits to reproduce and to lay their eggs. The introduction of apple tree provides a more nutrition food source for developing the maggots, as well as an escape from the parasitic wasp. The difference between the two types of fruits, such as maturation timing, allowed for the evolution of isolation mechanism and subsequent divergence of the two subspecies. The simplified flowchart of sympatric speciation: 1. Single interbreeding population. 2. Geographic isolation between the population. 3. Increase genetic divergence among the geographic isolation population by means of genetic drift and selection. 4. The selection for increased reproductive isolation. 5. Speciation completed. Parapatric speciation. The parapatric speciation. Para means beside, patrick's place. Parapatric speciation. The parapatric speciation, para behind, patrick place, is a form of speciation in which the evolution of a reproductive isolated mechanism occurs when the population enters a new niche or habitat within the range of a parent species. Generally, this occurs when there has been a drastic change in the environment within the original species habitat. In parapatric speciation, there is no specific extrinsic barrier to gene flow. The population is continuous, but nonetheless, the population does not mate randomly. The individual are more likely to mate with their geographic neighbor than with individuals in different parts of population's range. In this mode, the divergence may happen because of reduced genes flow within the population and varying selection pressure across the population range. Evolutionary force involved in speciation. The evolution is a process that results in the change in the genetic material of a population over time. The evolution reflects the adaptation of organisms to their changing environment and can result in altered genes, novel trait, and new species. Because the genetic change lies at the heart of speciation, the formation of new species involved the action of some evolutionary forces known to cause the genetic changes within the population. The principal evolutionary forces most commonly involved in species divergence or natural selection, genetic drift, and mutation. All these forces act to change allele frequency within the population over time, and there is no question that they operate during the speciation as well. Natural selection. It is a process by which the forms of organisms in a population that are best adapted to the environment increase in frequency relative to the less adapted forms over a few generations. Darwin was the first to popularize the idea that the speciation could readily occur through the prolonged action of natural selection, the process by which genetic variants that are better suited to the natural environment increases in frequency while variants that are not well suited decreases. Genetic drift. The genetic drift is the random change in allele frequency caused by sampling error across a generation in a finite population. The small population size can exert a strong force on the evolution of populations through genetic drift. While it occurs in all population, the drift can be a major driving force for changing allele frequency in the same population. If the small population or subjected to drift, the rate of evolutionary change can be speed up dramatically, and the alleles and genotype frequency can change unpredictable from one generation to the next. The smaller the population, the more extreme these fluctuation tends to be. The actual change in allele frequency caused by the genetic drift is random for any given generation. However, the effects of drift will accumulate over time. Much of the mathematical theory describing the process of genetic drift was developed by Wright. Like selection, the drift is a process of differential reproductive success. However, the key elements of genetic drift is that when individual survives and reproduce is random, unrelated to their phenotype and genotype. Principles of genetic drift, that is variation in relative genotype or allele frequency from one generation to the next, is the result of random sampling of gametes in finite population. If the population size is small or become reduced, a small proportion of the total gamete pool will end up forming individual in the next generation, and thus the genetic makeup of the population. Small samples taken from the genetic pool may not be representatives of range and relative frequency of alleles existing in the gamete pool. As a result, allele frequency may change dramatically from one generation to the next. The magnitude of these fluctuation tends to be larger in small population, and the effect of the drift or cumulative. Because it is a random process, the outcome change in allele frequency is any one generation is unpredictable. However, we can make certain generalities, and reliability predicts the cumulative effect of the drift. The genetic drift has several important effects on evolution. A. The drift reduces genetic variations in population, potentially reducing a population's ability to evolve in response to new selective pressure. B. It acts faster and has more drastic results in small population. C. It can contribute to speciation. For example, a small isolated populations may diverge from a larger populations through genetic drift. Certain circumstances can result in genetic drift having a significant impact on population. The two examples are founder effect and the bottleneck effect. Founder effect. When a small number of individuals known as founder populations becomes isolated from a larger population, this small group may establish a new populations whose gene pool differs from the source population. This is called the founder's effect. This is essentially a sampling difference. The larger the subpopulation, and more likely it will resembles the parent population. A classical example is the human population founded on a Pitcairn Island by several of Bounty mutineers and some Polynesians. The unique combination of a Caucasian and a Polynesian trial that characterize today's Pitcairn Island population resulted from the small number of founders of the population. Similarly, the Afrikaner population of a Dutch settler in South Africa is a descendant mainly from a new colonist. Today, the Afrikaner population has an unusual high frequency of gene that causes a Huntington disease, because those original Dutch colonists happens to carry the gene with the unusual high frequency. This effect is easy to recognize as in genetic diseases, but of course the frequency of all sorts of genes are affected by the founder's events. Genetic bottlenecks. Sometimes a population will be reduced to a very small number. The genetic diversity of a small population may be much less than the previous population, leading to a great change in allele frequency. An evolutionary event in which the population size is radically reduced is known as a genetic bottleneck. By chance alone, certain alleles may be overrepresented among the surveyor. Others may be underrepresented, and some may be absent altogether. The genetic bottleneck increases genetic drift, as the rate of drift is inversely proportional to the population size. Mutation. Neither selection nor drift can operate in the absence of genetic variability. Although quite a bit of variability can be generated just through genetic recombination and the random assortment of the chromosome during the reproduction. Ultimately, the spontaneous mutation is the primary source of genetic variations. The mutation is considered random with respect to the environmental challenges faced by organisms. Although that is not to say it is without bias, because mutations are important in the context of their effect on the phenotype of organisms. Certain phenotypic classes of mutations can be more common than other classes of mutation. Pattern of evolutionary changes. Most evolution consists of two processes: anagenesis, also known as a phyletic evolution, and a cladogenesis, also known as a branching evolution. The phyletic evolution is the progressive evolution of a species involving a gradual change in gene frequency in an entire population. When enough mutation reaches fixation in a population to significantly differentiate from an ancestral population, a new species named may be assigned. A key point is that the entire population is different from the ancestral population, so that the ancestral population can be considered extinct. When a species is transferred over time by the acquisition of a phenotypic and a genetic modification, there is no increase in the number of species. The cladogenesis is the evolutionary splitting event of gene pool into two or more separate pools, which give rise to one or more new species. The cladogenesis can promote biological diversity by increasing the number of species. A. Species 1 in percent of time to form species 2. Species 1 is divided into species 2 and species 3. Nature of evolution. Microevolution and macroevolution. The biological evolution occurs on different scales. These scales on which evolution occurs can be roughly grouped into two categories: the microevolution and macroevolution. The microevolution reflects changes in DNA sequences and alleles frequency within a species over time. These changes may be due to mutation, which can introduce new alleles into a population. In addition, new alleles can be introduced into the population by gene flow, which occurs during breeding between two populations that carries unique alleles. The evolution at this scale can be observed over a short period of time, for example between one generation and the next. The frequency of the gene for pesticide resistance in a population of pests increases. Such a change might come about because natural selection favors the gene, because the population receives new immigrants carrying the gene, because some non-resistant gene mutated to the resistant version, or because of random genetic drift from one generation to the next. One common example of microevolution is the size of the sparrow. House sparrow were introduced to North America in 1852. Since that time, the sparrow have evolved different characteristics in different locations. The sparrow populations in north are large bodies than the sparrow population in the south. This divergence in population is probably as a result of natural selection. The larger body birds can often survive longer temperature than smaller bodied birds can. Colder weather in the north probably selects for larger body birds. Hence, the sparrow in colder place are now generally of larger size than sparrow in warmer place. In contrast with microevolution, the macroevolution refers to a large scale evolutionary changes at or above the level of species, which results from the accumulation of numerous small changes on the microevolutionary scale. It means at least the splitting of species into two cladogenesis, or changes of species over time into another. Anagenesis, any changes that occurs at a higher level, such as the evolution of a new family, phyla, and or genera, and or all therefore macroevolution. The microevolution and the macroevolution are different things, but they involve mostly the same process. The microevolution is the change of allele frequency, that is genetic variation due to process such as selection, mutation, genetic drift, or even migration within a population. The macroevolution is an evolutionary change at the species level or higher, that is the formation of new species, new genera, and so forth. Despite their differences, the evolution at both levels relies on the same established mechanism of evolutionary changes, that is mutation, migration, genetic drift, and natural selection. Punctuated equilibria and phyletic gradualism. According to the theory of punctuated equilibrium evolution by jerk, the evolution proceeds relatively rapid during the speciation. Between the speciation events, the population remains relatively constant in a condition called stasis. The stasis is broken by rare events of large net change. The characteristics by a rapid event of branching speciation called a cladogenesis. It considered that most sexual reproducing species will show little change for most of their geological history. When the phenotypic evolution occurs, it is localized in a rare events of branching speciation and occurs relatively quickly compared to the species' full and stable duration on Earth. The theory of punctuated equilibrium is commonly contrasted against the hypothesis of phyletic gradualism, which states that most evolution occurs uniformly and by the steady and gradual transformation of whole lineage. Anagenesis, according to the phylogenetic gradualism. According to the phyletic gradualism, 1. Evolution occurs at a constant rate. 2. New species arise by the gradual transformation of ancestral species. Molecular phylogeny. The phylogeny is the science of estimating and analyzing the evolutionary relatedness among the various group of organisms. The molecular phylogenetic is a use of structure of molecules to gain information on an organism's evolutionary relationship. The nucleic acid, DNA, and RNA and protein are information molecules in that they retain information of an organism's evolutionary history. The approach is to compare nucleic acid or protein sequences from different organisms, either computer programs, and estimate the evolutionary relationship based on the degree of homology between the sequences. The nucleic acid and the proteins are linear molecules made of smaller units called nucleotide and amino acid respectively. The nucleotide differences within a genome or amino acid differences within a protein reflects the evolutionary distance between the two organisms. In other words, the closely related organisms will exhibit fewer sequence differences than distantly related organisms. Phylogenetic tree. In phylogenetic studies, the most convenient way of visualizing presenting evolutionary relationship among a group of organisms is through illustration called phylogenetic tree. The phylogenetic tree is represented by line and nodes. The nodes can be internal or external terminal. The differential sequence of DNA or protein which is compared are located at a external node but are connected via branches to internode, which represents ancestral form for two or more sequences. The branch defines the relationship between the sequence of DNA or protein in terms of descent and ancestry. The length of the branches indicates the degree of difference between the sequences represented by the nodes. The branch length are proportional to the predicted evolutionary time between the organism and sequences. The branching pattern of tree is termed as topology. A phylogenetic tree may be rooted or unrooted. The rooted tree interferes the existence of common ancestor and indicates the direction on evolutionary process. A rooted tree in which every node has two descendants is called a binary tree. An unrooted tree does not interfere a common ancestor and show only the evolutionary relationship between the organisms. Genetic tree versus species tree. A gene tree is a model of how a gene evolved through duplication, loss, and nucleotide substitution. As a gene at a locus in the genome replicates and its copy or passed onto more than one offspring, branching point are generated in a gene tree. A species tree depicts the pattern of branching of species lineage via the process of speciation. The speciation occurs by the population of ancestral species splitting into two groups that are unable to interbreed. An internode in a gene tree indicates the divergence of an ancestral gene into two genes with different DNA sequences, usually resulting from a mutation of one sort or another. An internode is in a species tree represents what is called a speciation event, whereby the population of ancestral species splits into two groups are no longer able to interbreed. These two events, mutation and speciation, do not always occur at the same time. Construction of a phylogenetic tree. The phylogenetic study constructs the tree-like pattern that describes the evolutionary relationship between the organism being studied. To construct a molecular phylogenetic tree, it is necessary to compare the nucleic acid sequence or protein sequence. A sequence of DNA yields more phylogenetic information than protein. The nucleotide sequences of a pair of homologous genes having a higher information content than the amino acid sequences of corresponding proteins, because mutation that results in a synonymous changes alters the DNA sequences but do not affect the amino acid sequences. Many ways of constructing the phylogenetic tree from aligned sequences have been developed, and numerous programs are available that performs the operation automatically, although some procedure constructs the alignment and the tree simultaneously. In more cases, the alignment is made first, after which the tree is calculated from it. Constructing a phylogenetic tree from molecular data is a complex process. There are three methods of tree constructions: the distance matrix method, maximum parsimony method, and maximum likelihood method. Currently, there is no perfect method. However, regardless of different methods being applied, the goal is same, which is to obtain a best estimate tree. Molecular clock. The molecular clock is a technique in molecular evolution to relate the time that the two species diverge to the number of molecular differences measured between the species DNA sequence or protein. It is sometimes called a GNA clock. It is sometimes called a gene clock or evolutionary clock. The concept of molecular clock is based on hypothesis that DNA and protein sequences evolve at the rate that is relatively constant over time and among different organisms. This consistency is used to estimate the length of the time that various organisms have been diverging from one another by measuring the degree of difference between two sequences. The molecular clock hypothesis was originally proposed by researchers Emil Zucker-Handel and Linnaeus Pauling on the basis of empirical observation, but it soon receives theoretical backing when biologist Moto Kimura developed the neutral theory of molecular evolution in 1968. Neutral theory of molecular evolution. The neutral theory of molecular evolution, also simply the neutral theory of evolution, is an influential theory introduced by Moto Kimura. The Kimura suggests that a large fractions of new mutation do not have an effect on evolutionary fitness, so natural selection would neither favors nor disfavors them. Eventually, each of these neutral mutations would either spread throughout a population and become fixed in all of its members, or they would be lost entirely in the stochastic process called genetic drift. Kimura then showed that the rate at which the neutral mutations become fixed in a population, known as a substitution rate, is equivalent to the rate of appearance of new mutations in each members of a population, the mutation rate. Provided that the mutation rate is consistent across the species, the substitution rate would remain constant throughout the tree of life. The neutral theory is the foundation of molecular clock technique. which evolutionary molecular biologists use to measure how much time has passed since the species diverged from a common ancestor. A strict molecular clock based on Kimura theory, neutral theory, is too simplistic because the rate of molecular evolution can vary significantly among the organisms. Thus, the researchers has undertaken effort to retain some aspect of a original clock hypothesis while relaxing the assumption of a strictly constant rate. Such efforts have led to the development of so-called relaxed molecular clock, which allows the molecular rates to vary among the lineages, albeit in a limited manner. Calibration of molecular clock. When using either a strict or relaxed clock method of genetic analysis, the most important consideration is how to calibrate the molecular clock. Assuming, for example, the researcher has two DNA sequences that have content differences of 5%. From this information alone, it is not possible to tell whether these sequences have diverged from each other at a rate of 1% per 1 million year over a period of 5 million year, or whether they have diverged at a five-fold higher rate over a period of just 1 million year. This is equivalent to trying to determine the average speed of car merely by looking at its odometer. To deduce the average speed, one would need to know the length of time for which the car has been traveling. Thus, to calibrate the molecular clock, one must know the absolute age of some evolutionary divergence event, such as a split between mammals and birds. An estimate of timing of this event can be gained by examining the fossil record or by correlating this particular instance of evolutionary divergence with some geological events of known antiquity, such as the formation of mountain range that splits the geographic range of species into two, thus initiating the process of speciation. Once the evolutionary rate is calculated using a calibration, this calibration can then be applied to other organisms to estimate the timing of evolutionary events. For example, fossils suggest that the most recent common ancestor of human and orangutans lived 13 million years ago. To calibrate the human molecular clock, we can compare human and orangutan DNA sequences to determine the amount of nucleotide substitutions X that has occurred, and then divide this figure by 13 followed by 2 to obtain the rate of substitution per million year. Hence, the number of substitution per lineage per million years is equal to x bar 2 into 13. Geological timescale. The geological timescale is a chronological sequence of a geologic and evolutionary event covering the physical information and covering the physical formation and development of Earth. The geological timescale is divided into eon, era, period, epoch, and age. Eon. The two or more geological era form an eon, which is the largest division of geological time, lasting many hundreds of millions of years. Era. Two or more geological period comprises an era, which is hundreds of millions of years in duration. Third, period. The period is a basic unit of geological time in which a single time of rock system is formed, lasting tens of millions of years. Epoch. An epoch is a division of a geologic period. It is the smallest division of a geologic time, lasting several million years. Age. An age is a unit of geologic time which is distinguished by some feature like ice age. An age is shorter than epoch, usually lasting from a few million years to about 100 million years. Mentalism. The chief difficulty was that the most prominent biologists of Mendel's time were not in habit of thinking in mathematical terms. Even the simplest term used by Mendel, even Charles Darwin, whose theory of evolution by natural selection depends on the genetic variation among the individual, failed to understand the significance of Mendel's finding. In fact, the Darwins performed breeding experiments on snapdragons similar to Mendel's on peas and got data similar to Mendel's, but they missed the point, still relying on the concept of blending. The Mendel's pioneering paper had no discernible influences on the scientific world for more than 30 years. Spontaneity of mutation. The Hugo de is one of the rediscovery of Mendel's work proposed by mutation, that is, changes in the phenotype of an organism occurs in just one generation, where the primary engine of evolutionary change. Hugo de Vries did his pioneering work in genetics using the evening primrose Onothera lamarckiana, which is now known for having sudden large mutation called macromutation in its overall phenotype. Hugo de Vries argued that these kind of mutations were the basis of changes in the phenotype, to which the Darwin Hugo de Vries argued that these kind of mutations were the basis for the changes in the phenotype, to which Darwin referred in the origin of species, and that therefore natural selection was neither necessary nor likely as a cause of evolutionary change. This mutational theory of evolution was accepted by most of the prominent geneticists at the turn of century and led to the widespread public testimonial that dwarfism was dead. This was, however, later accepted as false because the mutation or the source of variation in organisms, and Darwin's theory of natural selection is still valid. The mutation can occur spontaneously or they are induced. The spontaneous mutation are more important for evolutionary process. The details of spontaneous mutation are given in the chapter on mutation. However, this spontaneous mutation, also called a background rate of mutation, varies from species to species and from gene to gene. The measurements of the spontaneous forward mutation frequencies for various genes of phage and bacteria range from 10 to 8 to 10 to the power of 8 to 10 to the power of 10 detectable mutations per base pair per generation. For eukaryotes, estimates of forward mutation rates range from about 10 to the power of minus 7 to 10 to the power of minus 9 detectable mutation per base pair per generation. The mutation is the ultimate source of all genetic variation. It provides the raw material for evolution. Without mutation, all genes would exist in only one form. The allele frequency would not change, and thus the organisms would not be able to evolve, adapt to environmental changes. The mutation can be divided into beneficial, harmful, and neutral, depending upon whether they increases, decreases, or do not change the viability or fertility of their carrier. The viability and fertility in turn affects the overall fitness of the organism. Evolutionary synthesis. The rediscovery of Mendel's theory of non-breeding inheritance paradoxically made Darwin's theory seems even less likely, as a geneticist at the turn of the century believed that evolution occurs via mutation rather than natural selection. However, the founders of theoretical population genetics eventually united Darwinism, evolutionary theory, and Mendelian genetics in what is now known as a modern evolutionary synthesis. Ironically, one great unsolved problem in Darwin's master work on origin of species, Darwin and his later followers were faced with a seeming paradox. They describe evolution as a continuous, gradual changes over time, but species are distinct from each other, suggesting that some process has created a discontinuity of or gap between them. The credit for doing this most to crack this puzzle goes to Ernst Mayr, perhaps the greatest evolutionary scientist of the 20th century, along with Fisher, Dobbs, Hansky, Halden, Wright, Ford, Wrench, Ketverkov, Simpson, and Stebbins. Mayer achieved the modern synthesis in 1930s and 1940s that integrate Mendelian theory of heredity with Darwin's theory of evolution natural selection. The Julian Huxley coined the term when he published his book Evolution. The modern synthesis in 1942. The modern evolutionary synthesis is now providing a widely accepted account of evolution. It is also referred to as the new synthesis, the modern synthesis, the evolutionary synthesis, the millennium synthesis, and the neo-dwarfism synthesis. According to the modern synthesis, the genetic variations in population arises by chance through mutation, mistake in DNA replication, and recombination crossing over homologous chromosome during the meiosis. The evolution consists primarily of changes in the frequency of alleles between one generation and another as a result of genetic drift, genetic flow, and the natural selection. The speciation occurs gradually when population or reproductive isolated by geographic barriers. The modern evolutionary synthesis can be summarized in the following basic tenets. 1. The mutation, genetic recombination, natural selection, isolation forms the most important process of a microevolution. 2. All new trait arises due to the mutation, including the genetic recombination and changes in the chromosome numbers. However, the mutation only acts as a potential source of variability. 3. The natural source is the most vital process of a microevolution, and its role is more progressive and creative in bringing genetic variability. 4. The isolation, reproductive as well as geographic, forms the strong base of a formation of new species, that is macroevolution. The reproductive isolation to work effectively requires immense contribution to of mutation, natural selection, and geographic isolation. 5. The new species are formed by continuous action of a microevolutionary process over a long time span, cutting through a geographical span. According to the evolutionary synthesis, the evolution is the sum total of 5 essential evolutionary processes: 1. Gene mutation; 2. Changes in chromosomal number; 3. Genetic recombination; 4. Natural selection; 5. Reproductive isolation. The first 3 processes provides the essential genetic variability without which changes cannot take place. The last 2 processes guide populations of organisms into adaptive channels. In addition to the above, the 3 accessory processes affecting the working of evolution are: A. Migration; B. Hybridization; C. Genetic drift or change factor.